Thank you. So I'm very delighted to be here to talk about UniProt as a global hub of protein information on behalf of our colleagues at the UniProt Consortium and on behalf of the PI teams here, Alex Bateman at EMBO EBI, Alon Bridge at the Swiss Institute of Bioinformatics, and myself from PIR. So just a very quick outline, mission of UniProt Consortium, what we intend to accomplish, and then I'll drill down a little bit about expert curation and computational annotation, certainly the great challenge that we are facing with the growth of sequence data and the scientific literature. And so how do we go about sustain the curation activity really is a key question that we continue to ask ourselves and try to find the best answers and approaches to deal with it. And then a little bit about UniProt being a hub for omics integration and UniProt as a fair resource. And the last part, I'm going to focus on the impact. How do we measure impact of a resource, international resource like this, and sustainability regarding funding and funding issues that all the core data resources are actually facing. So again, UniProt is an international consortium. Our mission is to provide scientific community with this kind of comprehensive resource, high quality, freely accessible resource focusing on protein sequence and function. And again, international collaborations in Europe and in the US. This is the UniProt team. The PIs, the key staff, really work very closely with our entire team of the content curation experts. Our development team continue to adopt and advance our underlying computational informatics infrastructure to make our work more and more efficient and effective. And the entire team consists of nearly 90 FTEs, and certainly, it's a wonderful team, but also, a lot of sustainability that we have to deal with to continue to make sure that we have the most efficient operation as well in terms of the team. So UniProt, again, it's a resource that integrates sequence functional information. And so most people are most familiar with the flagship knowledge base. That's a UniProt knowledge base that consists of the SwissProt section and the Turnbull section. But we do have a number of other databases as well. The UniPark is the archive that bring in or import the sequence from external sequence resources. And then we also have other ways of organizing our sequence space to make it more efficient in terms of finding informations and also efficiencies for help getting functional information out. And that includes organizing the complete proteomes and also in terms of clustering sequence into sequence clusters based on different percentage of sequence identities, the UniRef 100, 90, and 50. So, so UniProt certainly it's really quite important that we balance our portfolio at different levels and to make sure that we do accomplish our mission. And so there are 5 major areas that we focus our activities on. Certainly, again, the expert manual curation, the computational annotation are foundational for us to get the sequence functional information into UniProt, but again, also providing a hub for many different types of omics information get integrated and easily navigated by our scientific community. Fair, so continue to look into ways how we can make our resource more findable, accessible, interoperable, and reusable, especially in terms of the reusability. That really is a key for us to increase the value, and how do we go about interface with the scientific community through various type of training and outreach activities. And so growth of UniProt sequences, certainly not surprisingly, the number of sequences continue to grow. And so we do have a way of trying to cluster our sequences. So the the curve on top, that's a UniProt, that's archive, and then we use against the reference clusters to reduce the sequence space through UniRef 190 and 50. And the bottom curve there, the light blue, that's the Trimble section, and the red line, that's the SwissProt section. As you can see that the most well-characterized section of the SwissProt remain relatively small, and the percentage actually is growing smaller because the sequence space continue to grow. And in addition to the growth of the sequence, certainly we are also faced the challenge of the growth of scientific literature, which we all witness that. And so how do we go about dealing scale? And that is really a balance between how much information we provide versus the growing sequence space. And so we have model organisms where we really provide the most rich functional annotation with experimental information. Then for other reference proteomes, provide some literature curation. And then, we have complete proteomes by similarity, and then there will be other sequences and metagenomic sequences which are essentially uncharacterized. And so we need to, in a way, try to deal with the scale by combining the two approaches together, both the expert manual curation and the automatic annotation. And so the expert curation of scientific literature, again, that's kind of the bedrock of what the UniProt is doing, is to, through this kind of literature curation, really synthesize and structure information on those experimentally characterized protein based on information that we can identify from the peer review article. And not only on the functional annotation, sometimes we also have to, through the scientific literature, to correct sequence. So we really ask ourselves, is the expert curation scalable? And so we spent a 10-month period, gone through quite a number of very carefully developed workflows for us to look at and examine our curation workflow to see where are the places where we have the most manual, that takes the most manual labor in order to do the expert curation. And is it possible for us to curate, how much do we need to curate in terms of the scientific literature that allow us to capture the most important key functional information. And so after this 9 months careful calibrated case study, we have published this paper last year in the Bioinformatics Journal on expert curation and scalability. And the conclusion using the SwissProt as a case study is that expert curation indeed is scalable. And that is quite reassuring in terms of sustainability of literature curation, in that, through literature triage, through much more efficient curation workflow, we will be able to sustain, to some extent, the literature curation. And so essentially, what our curators do is we evaluate about 50 to 60,000 papers a year. About 10,000 of those are curatable and added to UniProt. Others are deemed do not need to be curated because they are either redundant, low priority, not well supported, or out of scope. And so the sampling shows that about 90% of PubMed papers are out of scope. And so it's a study that allows us to really strategize how do we go about sustain our literature curation. But still, there are a lot of challenges. I think Christine previously mentioned several of these points here regarding challenges to sustainability, short-term duration of grants, and funding to create new resources. There's always a pressure to create new resources, but not to maintain existing resources. Competition against research proposal, that's where this research innovation needs to come in to support this kind of resources as well. And crowdsourcing has been discussed as a potential mechanism to solve the manual curation problem, but they are really issues that still are quite important issues to be addressed, and it's kind of overly optimistic. And however, we can still utilize that to some extent. And the last part is really there's a general lack of understanding the importance of expert curation. And so those are the kind of key issues that we really need to address as a community in terms of the curation community. And so in order for us to supplement information in the scientific literature, we also bring this so-called computational map, additional bibliography. So these are the papers describing the proteins entries of interest. However, those papers are not used by our curators to add certain functional lines or other information. And so they are just added as additional bibliography that relates to the entry that provide useful information. And so we add those articles by leveraging text mining tools. And many of those organisms, especially non-priority organisms, oftentimes those additional bibliography provide the only characterization information for those entries. Adding articles by leveraging curated sources. Again, there are many other curated resources, including model organisms and specialized resources. We try to bring in those scientific literature as well and add categorization so that the literature could be more easily navigated. Crowdsourcing. This is really a very limited crowdsourcing activity that allows us to work with authors of those additional bibliography directly to bring in those valuable literature information. Automatic annotation, again, it's important complement to the manual curation. About 36% of Trimble entries have this kind of functional annotation through especially in terms of names, enzyme, activities, interactions, subcellular location, goal terms, and so forth. About 74% we can add the sequence annotation in terms of sequence features, and then there will be a certain fraction, a small fraction there that are truly unvalidated and no further annotation is available. And this is supported by UniProt and SAR system where we have manually curated rules to do functional propagation and automatically generated rules. So in terms of UniProt as a hub for omics integration, again, it's really important that in order for us to move from genetic variation and all this desire for us to have better approach to precision medicine, it really requires us to know the role of protein function in the disease biology. And so that's what UniProt is trying to do, is to sit in that center of the hub, and we collect and collaborate with many resources, the best resources that provide biocuration in certain specialized areas, react on cellular pharmacological models, RIA, pathway reaction, intact, protein-protein interaction, PRIDE, proteome resource on the proteomics annotation and gene ontology and so forth. And so that allows us to really take this kind of community effort to bring the best functional annotation to the community through this kind of hub integration. And in terms of integration at the omics level, especially the importance of us to connect the genomic variation to functional annotation, again, very nicely demonstrated by Christine, in that we need to also have this kind of functional annotation directly presented in a place where most people would be consuming their genomic annotation information, and that is in the genome browser itself. And so we have this kind of direct linkage between the genome browser view and the functional annotation view. Likewise, with the mass spectrometry community, the specific isoforms, processing forms, modifications, those could be consumed together as well. And so this is a visualization interface on the website, but it's equally important for us to reach informaticians who can really take our tools to many, many different other applications. And so the protein APIs allow us to provide those complementary REST data services through the RESTful access and so forth. UniProt is fair. So in 2016, there was this scientific data publication. It's a commentary. They reviewed many different resources, and UniProt was used as an example of fairness and the resulting value added. And so, so certainly, in terms of dissemination format, not just text, but also RDF, that would be, again, directly consumable, the website and so forth. And so there are many different criteria that were reviewed, and so we provide this kind of fair access. Training outreach, again, a lot of activities in that area. And so I wanted to quickly just talk a little bit about the impact. So there are many ways to measure impact. So one can measure impact through citation. So this is, so we have this citation network to show how our UniProt paper describing UniProt get cited and used in this kind of web of science network. We can also demonstrate that because a lot of people, a lot of work, papers, publications, when they use UniProt, they don't cite the UniProt paper. Instead, they cite UniProt URL. So we can also measure the resource URL in addition to the publications for citation. And this is a great example that there's a specialized database, MobiDB. The access to the database grow. We see a tremendous jump there after they were cross-referenced. We provide reciprocal links to about 160 resources. Right after that, you see a significant jump. And also in terms of our links to external resources, continue to grow. And so one thing about measuring the impact is the reuse of the data, especially UniProt is reusing hundreds of tools and resources, including NCBI, but it's very difficult to measure how resources, to collect metrics as to how resources are reused. Resource organization also through Google Analytics, statistics, and we have that map to show it's utilized globally. FTP access, data transfer, unique visitors, impact the community served. So we wanted to know how UniProt actually are used by different biological, biomedical communities, and so we conduct a study to show all the different type of resources that cite the UniProt, how their distribution. So it's a way, again, for us to have better understanding of the user community. Now, as an ELIXIR core resource, again, those are the criteria. And I just wanted to use the last minute also to talk about the current funding. So, UniProt has been funded through the NIH U grant from NHGRI and some funding from IGMS. Certainly, EMBO EBI also have this EMBO core funding that is tremendously important to support EMBO EBI activity. PI, we have some NIH grants. Activity at SIB also supported by the Swiss federal government through SERI. And so the NIH funding contribute to about 40% of the entire UniProt consortium portfolio. And our annual funding at the present time is about 16 million annual. And again, the remainder of the 60% is through the EMBO and Swiss federal government. A little bit about the history of UniProt. So basically, it was 3 disparate resources. In 2002, the UniProt, the consortium brought together EBI-SIB and PIR. Rolf was the PI, and Amos Bayrock and myself was kind of the initial UniProt consortium when the PIs when it was started. Now, in terms of the new grant, this is the current status. So we apply to this Genomic Community Resource U24 mechanism. We apply for 5 years. Again, at the current funding level, 5.9 million per year. We receive a priority impact score of 14. But the final recommendation is funding for 3 years. And the funding, instead of coming from NHGRI primary and NIGMS co-funding, we have now co-funding from 4 additional NIH Institute, NIDDK, NHLBI, NIID, and NIMH. And the reason why they reduced the UniProt from 5-year to 3-year is because many of you are quite aware of this NIH strategic plan for data science. And so a new funding mechanism will be developed in terms of supporting this kind of data resources in the future. And so that would be a conversation that I think later on, during the panel, we can go into.
Okay, we're a couple of minutes into the tea break, but I guess we have time for one question if we have any. Yeah, Francis?
So how did the few, I mean, there are, I don't know how many NIH institutes there are, 18, 20 something? How come it's only those 4 that are part of this?
Only 4.
Yeah.
It took a lot of effort
Arm twisting.
to get those 4 on board, yes.
Okay. And so you did that or they did that?
We work very closely with RJ, who is the program officer from NHGI, and he's been tremendous in terms of making this all happen. But we have to discuss with every individual institute, identify areas of their particular interest, and then make this happen. Yeah, so it's a collaborative effort with NIH agencies.
Hi, I just wanted to pick up on the need for skills in biocuration, and also to kind of follow on from the discussion we had from Christine's talk, and do a rampant plug. So we have a new PG cert in biocuration that the EBI has developed in collaboration with the University of Cambridge Institute of Continuing Education, and the deadline for applications is the 31st of August. And the whole point of this is to provide some professional development for biocurators, yes, and to foster the idea that this is an important profession and a growing profession. So I hope that will be of interest to some of the people.
Absolutely, and again, the career advancement has been very much on top of the agenda of this, this ISB, right? International Society for Biocuration. Yeah.
And, well, they were actually instrumental in putting the curriculum together, as was Claire O'Donovan, who's also sitting in the room.
Exactly. And I think they are certainly in terms of our curation, with data growing so rapidly, and new technologies, new ways of looking at how you can best curate and integrate your data. So there will be a lot of advancement I think the biocurators could have that can really help them their career path as well. So yeah, it's wonderful. Thank you.
Thank you.
Okay, thanks. Let's take a tea break, and we'll be back here at 10:20. Yes. 10:15. Sorry.